Hello guys! In this part, we're gonna be talking about gene therapy. But before that, we need to define first what gene is. This is a gene. It can be found inside our cells, to be more specific, in the nucleus. It is a sequence of nucleotides in DNA or RNA. A gene is the basic unit of heredity. Some genes act as instruction to make molecules called protein. Many human diseases are caused by defective genes and improper protein functioning. Because chromosomes and genes provide instruction for our body, any changes to this structure have the potential to cause genetic diseases and negatively impact our health. These changes alters the product of gene expression, and the resulting protein does not perform its function as intended, which create problem to the cell and by extension to the organism. Let's take a look what a defective gene structure looks like. The gene expression on the left side is a normal gene that produces functioning protein, while the expression on the other side is a defective gene that produces protein containing tetratricopeptide repeat, which is involved in virulence-associated function of bacterial pathogens. What if we could fix defective genes? What if we could replace the defective allele with a normal allele? If the faulty gene can be replaced in every cell that contains it, then the disease will definitively be cured. And that is precisely what gene therapy seeks to do.
To say that your therapy has the potential to be curative is huge.
This is a particularly exciting time for gene therapy because the promise and the potential that has existed for 30 years in the field is now starting to finally bear fruit.
A small, microscopic virus called adeno-associated virus carrying a therapeutic gene can target specific cells to correct disease causing improper protein function. AAVs have a protective outer shell on them, which consists of viral proteins, and those proteins contains the molecular addresses to target specific cell types and tissues for delivery of the therapeutic gene. Once within the target cell, the outer shell called capsid is shed inside the nucleus to release its vector genome. AAV vector system can be administered by intravenous drip or direct injection to the target tissues.
Typically, AAV vectors are used to replace a defective gene that's found in a genetic disorder.
The real promise of gene therapy is to actually correct at the genomic level the cause of disease, which could in fact result in a lifelong cure without the need to take pills every day, to have painful injections every day.
Organisms inherit genetic information that result in continuity of structure and function between parents and offspring. Some genetic inheritance disorders are heart disease, high blood pressure, Alzheimer's disease, arthritis, diabetes, cancer, and obesity. Note that the therapeutic gene is not passed on to the future generation of the cells, so the harm of the disease to your offspring are still present. Because some individuals who are genetically positive for a certain disease may be entirely asymptomatic, this has been a problem for many couples. It is beneficial to know what chance their offspring have of inheriting one or several harmful genetic traits. There are many ways to know genetic materials inside us like PCR, Southern blotting, RFLP mapping, and DNA sequencing. But these are only providing us some information about our genes. They don't cure any genetic disorders.